Hi everyone, this is the first of three lectures on the respiratory system. The major functions of your respiratory system are to supply the body with oxygen for the process of cellular respiration and to dispose of the carbon dioxide, which is the waste product that is made through the process of cellular respiration. Respiratory and circulatory systems are closely linked or coupled to one another, and the respiratory system also functions in olfaction and speech. So the process of respiration is actually four different activities that occur. The first one is called pulmonary ventilation, which you and I know of simply as breathing. This is the physical movement of air in and out of your lungs. Then you have what's called external respiration, which is the exchange of the oxygen that is in the lungs into the blood and the carbon dioxide that is in the blood into the lungs for exhalation. Then you have the transportation of gases in the blood. And then finally, you have internal respiration, which is the process of exchanging the oxygen that is in the blood with the carbon dioxide that's in the tissue. So dropping off the oxygen and picking up the carbon dioxide and carrying that carbon dioxide back up to the lungs and breathing it out. So the process of pulmonary ventilation and external respiration is carried out by your respiratory system, and the process of transportation and internal respiration is carried out by your circulatory system. So let's look at a little bit of functional anatomy or a review of the anatomy structures. So the major organs that we'll be talking about are the nose and the nasal cavity, the paranasal sinuses, and the pharynx. And those three sets of structures make up what is referred to as the upper respiratory system. And then you have the larynx, the trachea, the bronchi, and its branches, as well as the lungs and the alveoli. And all of those structures are considered part of your lower respiratory system. So basically, at the voice box, at the top of the voice box is where the transition between upper and lower respiratory occurs. So when your patient has an upper respiratory infection, it's something going on in those structures up at the top. And if it's got a lower respiratory infection, it's something from the voice box down. So there are some pictures just of the different structures. And then you've got some tables that tell you the names of the structures, what they do, and where they, it's just a general description of them. And then there's lower respiratory. So let's start with the upper respiratory system and talk about these structures. So we'll talk about the nose and the paranasal sinuses first. So the nose is the only portion of your respiratory system that is actually external, that we can actually see, right? The function of your nose has lots of functions. It provides an airway for respiration. It moistens and warms the incoming air. It filters and cleans that air as well. It is a resonating chamber for speech. Some people are real nasally when they talk. If you ever saw the TV show The Nanny, Fran Dressler plays a part in there where she talks through her nasal cavity. And it also houses the receptors for your sense of smell. The nasal cavity is divided into, or the nose is divided into two regions, the external nose and the nasal cavity. So the external nose has different areas of it. The root is the portion of it that is between your eyebrows, and then of course as it starts to jut out, that's referred to as the bridge, and then the dorsum nasi is the anterior margin or the long part of your nose and down on the sides, and then the apex is the tip of your nose. The nostrils are the openings from the outside, and they are bound on the lateral sides by the ala. The external nose is made of the nasal and the frontal bones. Those are what form the bridge and the root respectively. The maxilla bones on the sides, and then some plates of hyaline cartilage on the tip. So there are the different structures that I was just talking about of your nose. And then a close-up of that, looking a little more closely at the different cartilages that make up the tip of the nose. The nasal cavity is what's inside of your head. And so it is divided down the middle by the nasal septum, and the septum is made of cartilage, and that cartilage is attached to the vomer bone and the perpendicular plate of the ethmoid bone. So if you remember back to 210 when we were looking at the skull, the ridge that's in the middle of the nasal cavity that empty ridge is where the septal cartilage would sit. All the way at the back of the nasal cavity, you have the posterior nasal aperture. Some people still refer to those as the internal nares. That's the opening where the nasal cavity turns into the nasopharynx, or the first part of the throat. There's the roof, which is formed by the ethmoid bone and the sphenoid bone. The floor of your nasal cavity is the roof of your oral cavity, so it's made of the hard palate and the soft palate. So the hard palate is the bone portion that we'll talk about, and the soft palate is where there's no more bone and it's just muscle. The nasal vestibule, this is superior to the nostrils. So don't do this, but you could stick your finger in your nose and you notice that it goes, your finger goes, you can make your finger go up. And that's the nasal vestibule. It's lined with hairs that are serving to filter out coarse particles from anything that you are inhaling. The rest of the nasal cavity is lined with mucous membrane. And so there's some changes in that we want to talk about. So the olfactory mucosa is what lines the very top of the nasal cavity and contains the structure that we talked about at the very beginning of the semester, or the very end of last semester, which was the olfactory epithelium, which is the tissue that's responsible for our sense of smell. And then you've got the respiratory mucosa, which is pseudostratified ciliated columnar epithelium that has lots and lots of goblet cells in it, and it's sitting on the lamina propria that has many seromucous nasal glands. Those seromucous nasal glands have mucus secreting mucus cells as well as serous cells, hence the name of it. And the serous cells are secreting a watery fluid that has lots of enzymes. The mucus and the serous secretions also have lysozymes and defensins. They have to be able to protect you from anything microbial that is in the inspired air. Ciliated cells are going to sweep the contaminated mucus back towards the throat, and then we swallow that down into our stomach, and the stomach acid kills any of the bacteria that's in there. Inspired air is warmed by the capillaries that are very close to the surface of the epithelial tissue on the inside of your nose. You might have experienced nosebleeds if you've ever your nasal cavity has been really, really dried out, like maybe you have a real bad cold or allergies and you're taking Benadryl and it really dries out your nasal cavities, and you know how easy it is for you when you're blowing your nose to get a nosebleed because the capillaries are so close to the surface. But that's what's warming that air as it's coming in, and it's very efficient. If you have ever been someplace that's really, really cold, and you try to breathe in through your mouth rather than breathe in through your nose, the cold air actually hurts your lungs. So this warming of air as it's coming in is a really important idea, which is why you want people to breathe through their nose and not through their mouth. And then finally, the nasal mucosa contains a lot of sensory nerve endings that can cause sneezing to force out anything. So what sneezing is a reflex that helps us to expel anything that we breathe in. So at the time that I'm recording this, there is a lot of pollen in the air, and so lots of people are walking around sneezing. That pollen is getting into their nasal cavity, and their nasal cavity is very, very sensitive to particulate of any type, and it's causing them to sneeze. The nasal conchae. So I want you to try to remember back to 210 where we talked about the nasal conchae, and there were three of them on each side, and they were like little shelves of bone that were on the lateral side of the nasal cavity. And so they say they're scroll-like, and they're covered by mucosa, and they protrude into the nasal cavity. So the old name for the nasal conchae were the turbinates. And that word is probably more fitting because what it does is causes an increase in the turbulence of the air. So as the air comes into your nose, it's kind of swirled around, and as it's swirled around, it gives an opportunity to be warmed and humidified and filtered and all those kinds of things. All right. Three of those nasal conchae that we talked about last semester, the superior, the middle nasal conchae, which were markings of the ethmoid bone, and then the inferior nasal conchae, which was a bone all by itself. The nasal meatus is the groove in between each of those. So we name them according to the one below. So there's a superior nasal meatus, a middle nasal meatus, and an inferior nasal meatus. And the shape of it, as I mentioned, helps to increase the surface area as well as cause the air to become very turbulent and swirl around inside of your nasal cavity. During inhalation, the conchae and the nasal mucosa function to filter heat and moisten the air, as I mentioned a few minutes ago, and during exhalation, they help to reclaim some of that heat and moisture. The paranasal sinuses make a ring around the nasal cavity. These are open spaces that are in the bone, and they are named according to the bone that they are found in. So there are two of them for each. There's the frontal, the sphenoid, the ethmoid. Some books will tell you that the ethmoid sinus is called the ethmoid air cells because it's actually a whole collection of little air sacs inside or air spaces rather than one open space. So collectively, they call it the ethmoid sinus and the maxillary bones. The function of these sinuses are to lighten the skull so that your skull is a little bit lighter. Secrete mucus, which is where the sinuses get some of us in trouble, and to help to warm and moisten the air. So there are the sinuses. Again, there are two of them. You only had one frontal bone, but there are two frontal sinuses. They're showing you where the ethmoid sinuses are, or the ethmoid air cells, as I mentioned, the sphenoid sinuses, and then the maxillary sinuses, and then a side view of those as well. So you can see why with the ethmoid, if you're looking at side view B, you see the ethmoid is actually different whole bunch of little cells, and that's why they call them ethmoid air cells in some books. And they're taking a look at a cross-sectional view. So you can see those three nasal conchae, superior, middle, and inferior, and the meatus in between them, as well as the sphenoidal sinus, the frontal sinus, those kinds of structures. There it is looking at it from a cadaver. So rhinitis is inflammation of the nasal mucosa. The nasal mucosa is continuous with the rest of the respiratory tract. So if you've ever had like an inflammation or a cold in your nose and you notice it travels down your throat and eventually ends up in your chest, it's because it's all connected. It can also spread to your tear ducts and to the paranasal sinuses and cause a sinus infection. It can lead to the absorption of air, which produces a vacuum, and that's what gives you a sinus headache in those paranasal sinuses. Moving along down the tube is the pharynx. So this is a funnel-shaped muscular tube that runs from the base of the skull down to about C6. It connects the nasal cavity and the mouth to the larynx, which is your voice box, and the esophagus, and it is made of skeletal muscle. There's three regions of the pharynx that are named according to where they are. So the nasopharynx, oropharynx, and laryngopharynx. And so this picture does a nice job of showing you those three regions in different colors. So the area behind the nose down to the tip of the uvula, that's what that little pointy thing is that we'll talk about, is the nasopharynx, and that's in red. And then from the uvula down to the epiglottis, which is blue, the blue area is called the oropharynx, and then the area behind the larynx is called the laryngopharynx. So the nasopharynx is the air passageway from the nasal cavity into that area. So it is lined with pseudostratified ciliated columnar epithelial tissue. The soft palate and the uvula are going to close during swallowing and make it so things can't back up into the nasopharynx. So the uvula is the structure you see when you open your mouth and you look in the mirror and there's a hangy thing down in the very middle. That's the uvula. And so the soft palate is what it's attached to. And when you swallow, that seals up. And so when you swallow, it directs things down your throat rather than back up through your nose. You also have the pharyngeal tonsils, which we've already looked at, better known as the adenoids, that are located on the posterior wall. Some people say there's two, some people say there's one. We kind of argue about that all the time. And then the pharyngeotympanic tubes, which were the auditory tubes from the ear. Remember the tube that was coming off the middle ear, draining down into the throat. That is where those structures, the openings to those are. There's some little tubal sinuses or tubal tonsils located around those auditory tubes where they enter into the nasopharynx, and they help to drain and equalize the pressure in the middle ear, and they open into the lateral walls. We talked about those last semester in Bio 210. The middle ear, if you remember, is supposed to be air-filled, and so those openings right there is how fluid backs up into that middle ear and causes otitis media. The oropharynx is the passageway for food and air from the level of the soft palate to the epiglottis. So basically the hangy thing, the uvula, and down to the epiglottis. So this is stratified squamous epithelium. So I want to go back to the tissues chapter in your mind, and I want to remind you that whenever we had stratified tissue, its number one function was protection. So the oropharynx and the oral cavity, we're constantly eating food and drinking things, and that tissue, those cells are abraded away from the surface. So if we only had a single layer of cells in the oral cavity and the oropharynx, that as soon as we ate something, those would be abraded down to the next level, and we wouldn't be protecting the tissues at all. So stratified squamous epithelia happens to be non-keratinized as well in this particular area. The isthmus of the fauces is the opening to the oral cavity. The palatine tonsils are the ones that you think of course, when someone has their tonsils removed. They're the ones that are on the lateral walls, and then the lingual tonsil is the one that's found on the base of your tongue. The laryngopharynx is the last little chunk of the pharynx, which is a common passageway for both food and air. It is posterior to the upright epiglottis. So when the epiglottis is open, so in other words, you're not swallowing food or water at that particular time, extends to the larynx where it becomes continuous with the esophagus. It is also lined with stratified squamous epithelial tissue that is non-keratinized. So looking at those different regions and some of the structures that I was just talking about. So infected and swollen adenoids can block the air passageway in the nasopharynx and making it necessary to breathe through the mouth. So this is where you get a lot of times problems with mouth breathers. As a result, the air is not properly moistened, warmed, or filtered before it reaches the lungs. When the adenoids become chronically enlarged, both speech and sleep can be disturbed. So they'll go ahead and remove those. They'll usually remove the palatines at the same time. Moving right along, we'll go down to the lower respiratory system. So the lower respiratory system starts at the larynx, includes the trachea, the bronchi, and all of its branches, as well as the lungs. So it's broken into two zones. The respiratory zone is where gas exchange is happening, and this is all the way down at the very base. So this is the respiratory bronchioles, the alveolar ducts, and the alveoli, so down all the way deep in your lungs. So these are microscopic structures. The other portion of it is called the conducting zone, which is basically the tubes that are transporting the gases, air, to and from the exchange surfaces. So this is everything else when we start talking about the lower respiratory system. So the majority of it is the conducting zone. So it cleans, this is the area that also cleanses, warms, and humidifies air as it's going down. So we'll start at the top with that, with the larynx. The basic anatomy of your larynx, better known as your voice box, is that it is sitting between the third and sixth cervical vertebrae, and it is attached to the hyoid bone. So if you remember the hyoid bone, it was the only bone that had the distinction that it didn't articulate with another bone, and basically what it does is it stabilizes your larynx. The larynx opens into the laryngopharynx and is continuous with the trachea, so it's basically at the top of that, of the trachea. There's three functions of the larynx. It provides a patent airway. Patent means it's always open. It routes air and food into the proper channels. So we want to make sure that air goes down the trachea and food and water go down the esophagus. And it's also responsible for the production of your voice, which is phonation. It houses your vocal cords. So the larynx is made of 9 cartilages. 8 of those are hyaline. 1 of those is elastic. So it is 9 cartilages, but they're not all hyaline. It's connected by membranes and ligaments. The biggest one that you see on the very front is called the thyroid cartilage. Sometimes people call that the Adam's apple, and the ridge right up the very front of it that's very obvious on males after puberty is called the laryngeal prominence or the Adam's apple. The cricoid cartilage is right below that. It's ring-shaped, so that's cartilage number 2. Cartilages 3 and 4 are a pair called the arytenoids. So when you look on the back side, there are these two structures that look like the talons of a hawk. Those are called the arytenoids, and on the very tips of those are the corniculates. And then embedded over in the side membrane are the pair of cuneiforms. And then the 9th cartilage is your epiglottis, the one that's made of elastic cartilage. So again, it is not hyaline. It covers the laryngeal inlet during swallowing. In other words, it's kind of like a little manhole cover. It flips down over the trachea because the trachea, remember, is patent. It's always open. So it flips down over top of the larynx when we're swallowing food or water and keeps things from going down the wrong tube. It actually has taste buds in the mucosa. And so if you've ever, if somebody has maybe sprayed perfume or some chemical, like maybe cleaning solutions or something near you, and when you inhale it, you feel like you can taste it, that's exactly why. So showing you there where the hyoid bone is, where the laryngeal prominence is, where the cricoid is, and giving you some reference points there with the sternocleidomastoid, as well as the clavicle and the jugular notch. So there is a picture from the front view of the structure I was just talking about. So the vocal folds, the vocal ligaments are what form the core of the vocal folds, or your true vocal cords. The ligaments are deep to the laryngeal mucosa, and they attach from the thyroid cartilage on the front straight back to the arytenoids on the back. So they're kind of running through the very middle, down the middle of it. And they have elastic fibers that look white because they do not have any blood vessels. The opening between the vocal folds is called the glottis. So we've been using the term lumen when we were talking about vessels, and we're going to use the term lumen later on as well. But at the larynx, the part down the very middle of the structure, the empty part, is called the glottis. So that's why the structure that's sitting over top of it is called the epiglottis. The folds vibrate and they produce sound as air rushes up and down from the lungs. So our inhale and exhaling that we do, that's connected to our speech. The vestibular folds are called the false vocal cords, and they are superior to the vocal folds, or out to the side really, and they don't have anything to do with the production of sound. What they do is to help the glottis close during swallowing and make sure that nothing falls down the glottis. So even if you get a little teeny tiny piece of dust on your false vocal cords, it makes you cough like crazy. Right? It makes you run tears and you can't stop coughing. It shows you how protective those vestibular folds are. The epithelium of the larynx, the superior portion of it is stratified squamous epithelium, and then inferior to the vocal folds is pseudostratified ciliated columnar epithelium. So taking a look there, so we're looking at this from the back. So just to kind of get you oriented, you can see the glottis right down the very middle there, and they're showing you the arytenoid cartilage that looks kind of like the talon, I said, like of an eagle or of a hawk, and then on the very tip of that is the corniculate. This picture is really good for seeing from the side, seeing the one cartilage that you can't see. And that's the cuneiform, or the cuneiform, I guess is probably the more correct way to say that. And so that is going down the side right there. It kind of looks like a nail right through the middle of the pink muscle that you see there. So this picture is showing you, this is as if we were looking straight down someone's throat. All right, and so the picture on the left, if you look in the very middle, you see those two white bands. And those two white bands, those are the vocal cords, the true vocal cords. Okay. And then on either side of that, that tissue going on either side, that is going to be your false vocal cord. And so on the picture on the right, you can see when that's open. So again, the true vocal cord are those bands that are on either side of that. And then all the tissue going out past that to the sides, those are going to be the false vocal cords. So how is voice produced? Speech. Speech is actually the intermittent release of expired air during the opening and closing of your glottis. And so the pitch is determined by the length and the tension on the vocal cords. Loudness depends upon the force of air. So when you need to talk louder, you take a deep breath and you really can put your sound out there. The chambers of the pharynx and the oral and nasal and sinus cavities amplify and enhance the sound quality, and the sound is sharpened into language by muscles of the pharynx, the tongue, the soft palate, and the lips. So the pitch idea is really interesting. If you've ever been around a little one when they're learning to talk, and they make a lot of squealy noises at first. Basically, what their brain is doing is figuring out, oh, when I have this tension on the vocal cords, it sounds like this, and when I have this tension on the vocal cords, it makes this sound. And so they're practicing that. I mean, they don't consciously realize they're doing that, but that is part of the process of them figuring out how it all works. The sphincter functions of the larynx, the vocal folds may act as a sphincter. A sphincter is just a ring of muscle that's controlling movement, and what it does is it prevents air passage. An example of this is the Valsalva maneuver. This is when the glottis closes to prevent exhalation and the abdominal muscles contract and the intra-abdominal pressure rises. So this is anytime you're told to push. So if you have had a baby and you're pushing that baby out, you're doing the Valsalva. People do it all the time when they empty their bowel, but they also do it when they stabilize their trunk when they're picking up something really, really heavy. You basically push down and from your throat, and that's a Valsalva. Laryngitis is inflammation of the vocal folds, and that causes the folds to swell up, and it interferes with the ability for them to vibrate. So you sound very squeaky if you have any voice whatsoever. So it's going to cause changes to your vocal tone. It can make you sound very hoarse, and in some cases, you might not be able to get any sound out whatsoever. Laryngitis is most often caused by a viral infection, but it can be because of overuse. So, back years ago when I very first started teaching, when the beginning of the semester would roll around and I had to do a lot of talking at the beginning of the semester and I hadn't had to talk for a couple of weeks between semesters, I would always end up with laryngitis. I guess now I've just worn my poor vocal cords to the point where they're calloused and hardened, and that doesn't happen anymore. Laryngitis also can happen when you have really dry air. It can be from a bacterial infection. It can also be from tumors. So you sometimes hear about that, especially with singers, because that's part of what allows them to hit the notes that they can hit. Um, and you can sometimes get laryngitis from inhalation of irritating chemicals or pollen or things like that as well. The trachea. The trachea is also known as the windpipe, and it goes from the larynx into the mediastinum, which is the space between your lungs, and it divides at that point into the two main bronchi. So it's about 4 inches long. It's about 3/4 of an inch in diameter, and it's very flexible. It's kind of like the bendy part of a straw. There are walls of this. So the innermost wall is called the mucosa. It is ciliated pseudostratified epithelium with lots and lots of goblet cells. And then going outward, we have the submucosal layer, which is made of connective tissue with lots of seromucous glands. And it's in this area that we find the C-shaped cartilages that keep the ring, that keep the trachea from collapsing. So they're called C rings, or tracheal cartilages. And then the outermost layer is connective tissue, and it's called the adventitia. On the back side of the trachea, you have something called the trachealis, which is smooth muscle fibers that connect the back side of the cartilage rings, because the rings are C-shaped. They're not O-shaped, they're C-shaped. So they're open on the back side, and what connects those two sides is the trachealis, and that is what contracts during coughing to help expel the mucus that is in your trachea. The carina looks like a little pair of bikini bottoms, and this is at the very end of the trachea. It's the last tracheal cartilage, and it is where it splits, the trachea splits into the two main bronchi. The mucus of the carina is highly, highly sensitive. And so if anything touches that, if somehow or another you manage to get something down there, it causes violent coughing to try to expel whatever it is that's down there. So showing you a cross section of the trachea. So you see the lumen. So now we've gone from um, going back to using that term lumen to mean the opening down the middle. We used glottis when we were up in the trachea, in the larynx, but we call it the lumen again. And you see that the light blue structure is shaped like the letter C, lying kind of in a funny orientation. And you can see that it doesn't go all the way back. And then you can see what the trachealis is what connects the two open ends of the letter C. And then you see that other tube that's right behind that, that is your esophagus. So the esophagus is behind the trachea. Picture here showing you the tracheal wall, so you get the idea of the pseudostratified ciliated columnar epithelium, and then the lamina propria that's made of connective tissue, and then we have the submucosal layer. So those two things I just talked about were the mucosal layer. Then you have the submucosal layer, which is got lots of seromucous glands, and then you have the hyaline cartilage below that. So scanning electron micrograph of the cilia that you find in the trachea, they are very, very abundant. And what do? Those cilia, they are constantly in motion. And so this little animation that you see here, the big green circles, those are goblet cells, and they are secreting lots and lots of mucus. And mucus is heavy. So mucus falls down your trachea. The function of mucus is always to try to trap particulate and obviously that little purple guy, that little microbe there. And so what the cilia do is they sweep upwards. Notice that they're sweeping upward, and that mucus that was really heavy and trying to fall into your lungs gets swept upwards, and you either expel it by spitting it out or you swallow it down into your stomach, and your stomach acid takes care of whatever's in there. So this whole idea of how this is working is called the mucus escalator. When you smoke, you burn those cilia off. And so the um, mucus travels down towards your lungs and you have to manually move it back up. And that's what a smoker's cough is. So when somebody's coughing when they've been smoking, not when they're actually smoking, but afterwards, what they're doing is they're trying to move that mucus up. If you stop smoking, those cilia grow back in 14 to 21 days. So again, smoking inhibits and ultimately destroys the cilia. And without that, coughing is the only way to keep mucus from accumulating in your lungs. And so this is the reasons why smokers with respiratory congestion should avoid medications that inhibit their cough reflex. So there's several different medications that they give you when you've got some type of a respiratory infection, and one of them is to keep you from coughing all the time. Well, if your patient is a smoker, they shouldn't be doing that because they need to cough to get that mucus up. Tracheal obstruction is life-threatening, so lots of people have suffocated after choking on a piece of food that suddenly closed off their trachea. We use what's called the Heimlich maneuver, which is the procedure in which air in the victim's lungs is used because we force it out, and it expels the obstructing piece of food. So the maneuver is easy to learn, but it's best learned by demonstration. So when it's done incorrectly, you can crack somebody's ribs, or you can cause that cracked rib to get stuck into their diaphragm or push through and go into their liver if you're not careful. So you want to make sure you know how to do it correctly. So now let's talk about the bronchi and the subdivisions. So the air passages after we get to the end of the trachea undergo 23 orders of branching. So before you gasp and think you have to learn 23 steps, you don't. but there are 23 different branching patterns. And so that whole thing is called the bronchial tree because if you flip it upside down, it looks like a tree. From the tips of the bronchial tree, the conducting zones are going to give rise to the respiratory zones. So let's start with the conducting zones. So the trachea stops at the carina. The carina, again, is the last C ring that looks like a little bikini bottom. And it forms the right and left primary bronchi, or main bronchi is the other term some people use. The right main bronchus is wider and shorter and more vertical than the left. Each main bronchus enters at the hilum of the lung. So the hilum is always the indention where everything goes in and goes out. Each main bronchus then branches once it gets inside the lung into the secondary bronchi, also known as the lobar, because each one goes to a lobe. So you have 3 lobes on the right-hand side of your lung chest, and you have 2 lobes in your left lung. So the secondary bronchi on the right side, there are 3 of those, because 1 for each lobe. And on the left side, there are only 2. And then inside of the lobe, the lobar bronchus branches into the segmental or the tertiary bronchi. And these branch a whole bunch of times. So this is part of that 23, right? The bronchi become smaller and smaller. When they are less than 1 millimeter in diameter, they're called a bronchiole. And then it's ultimately going to lead down to the terminal bronchioles, which are the smallest of the branches, which are less than 0.5 millimeter in diameter. So showing you that pattern, so we're looking at the blue stuff. Notice again that the lung on the right side has 3 lobes, and then the lung on the left side has 2 lobes. So we have the trachea, then you get to the bottom of the trachea and you see the bikini bottom, right? And so that's going to be the carina, and then that's going to branch, and those are going to be the right and left primary bronchi. As soon as they enter into the lungs, they're going to split into the lobars, one going to each lobe. And then once they are in the lobes, they're going to split again and become the tertiary bronchi within the lobe. In the conducting zone from the bronchi to the bronchioles, there are lots of changes that are going to occur. And that's what helps us figure out where we are in the system. So the support structures change. So we started out with C rings or cartilage rings, and they become more irregular, and they're more like little patches. In the bronchioles, elastic fibers completely replace the cartilage altogether. The epithelial tissue changes as we move through. So we had pseudostratified ciliated columnar in the trachea, which gives way to cuboidal, which ultimately, when we get all the way down, is going to become simple squamous. But in the conducting zone, it goes from pseudostratified columnar to cuboidal. The cilia and the goblet cells become less numerous the further down we go. And the amount of smooth muscle increases. So this allows the bronchioles to provide resistance. So we talked about resistance with blood vessels, and when we change the diameter, we change the resistance. And the same idea is going to be happening with the bronchioles. In the respiratory zone, this begins where the terminal bronchiole feed into the respiratory bronchioles, which lead into the alveolar ducts, which lead into the alveolar sacs, and the individual structures that are found on the alveolar sacs are called the alveoli. You have approximately 30 million alveoli. This is what makes up the most of the volume of your lung, and this is where the actual gas exchange happens. So up to this point, we were just getting everything down to the place where the gas exchange could happen. So taking a look here, so you've got a bronchiole, that's the one that's not labeled there, and then you see where the terminal bronchiole is, then you see the respiratories when it breaks into when it splits, and then once it enters into the part that looks like a Q-tip, that tube becomes the alveolar duct, and the little individual white cotton balls on the outside of there are the alveoli, and a whole bunch of them together is called the alveolar sac. So there we are looking at the inside of the lung histologically. So you're looking at a respiratory bronchiole over on the left-hand side and then the alveolar duct, and then you're seeing the individual alveoli. So it's important to realize that the alveoli are not complete circles. They all are half circles that open up into that duct running down the middle. The respiratory membrane. So there is a blood-air barrier that consists of the alveoli and the capillary wall. Because remember, the capillaries are wrapped around the alveoli because that's where gas exchange is going to happen, right? Remember back to blood flow through the heart. The blood goes out to the lungs and exchanges the carbon dioxide for the oxygen, heads back to the heart. Well, where does that exchange happen? It happens at the alveoli, and it happens because the respiratory structure has the capillaries, the blood structure, wrapped around the outside of them. So that's an interface between two systems that you're seeing there, between the respiratory system and between the blood supply. All right. The uh, basement membrane of the simple squamous cells in the alveoli and the basement membrane of the simple squamous cells in the capillary, because they're both got simple squamous lining them, the basement membranes fuse together, and that is what we call the respiratory membranes. Very, very thin. We need it to be thin because gas exchange is happening by simple diffusion. So if you remember all the way back to 210, diffusion only happens over short distances for the most part, and it has to be through a very, very thin membrane because we're not using any energy for this to happen. So this membrane can't be thick at all. The alveolar wall, again, is made of a single layer of simple squamous cells, what we call type 1 alveolar cells, and then scattered between those cells are type 2 alveolar cells, which are cuboidal, and they are secreting a product called surfactant. So surfactant is what keeps the lung from collapsing. And they also are secreting antimicrobial proteins. So if somehow or another some pathogen managed to make its way all the way down into your alveoli, there actually are proteins down there to try to kill it. Some other features of the alveoli, they are surrounded by fine elastic fibers. We want lots and lots of recoil in this area, and of course by those pulmonary capillaries that I was talking about a few minutes ago. The alveolar pores connect the adjacent alveoli to one another, so they have little holes in between them. That's what allows them to equalize pressure throughout the lung and provides an alternative route in case there's some type of blockage in the alveoli itself. And then you've got these alveolar macrophages, what they used to call dust cells, and they're keeping the surface of the alveoli sterile. So you have 2 million dead macrophages per hour that are carried by the cilia to the throat and swallowed or spit out. So again, air is not sterile, you know that, and even as good of a filtering process as we try to use with all the mucus and the hairs and all those things that happen in the upper respiratory system, there are still microbes that make their way all the way down. So 2 million of them an hour that our alveolar macrophages take care of for us. So there is a gigantic picture of the relationship between the capillary and the alveoli. So you notice again with capillaries, we talked about this already. Anytime you look at a capillary and you see red to purple to blue, you know that gas exchange is happening in that particular area. So this is the site of external respiration, the exchange between the oxygen and the carbon dioxide. The oxygen that's in your lungs and the carbon dioxide that is in the blood vessels has to be exchanged, and the oxygen goes into the blood and is carried down to the tissues, and the carbon dioxide goes into the lungs and is exhaled. Looking at a micrograph, a scanning electron micrograph of the pulmonary capillaries. And then just taking a close-up of the inside. So again, the alveoli are not solid circles. They are half circles. So if you could imagine what this picture is, plate C, it's almost like if you could imagine if you could walk down the middle of the alveoli duct, this is what you'd see on the walls. You'd see the inside, the half circle of the alveoli. They're also showing you the respiratory membrane on that last slide. Now let's talk about your lungs and the pleura that they are associated with. So the gross anatomy of your lungs, so they occupy all of the thoracic cavity except for the mediastinum. The root is the site of vascular or blood supply and bronchial attachment. to the mediastinum. The costal surface is what is the other surfaces, the anterior, lateral, and posterior surfaces. They are in touch with your ribs, and so the word for ribs is costal. And so the other parts of those are named according to which part of the ribs are they touching. The apex is up at the top, unlike we saw with the heart, which it was down at the bottom. It's underneath of your clavicle, and the base is what's sitting on the diaphragm. The hilum is on the medial surface or the mediastinal surface, so it's on the medial aspect of the lung, and that's where the entry and exit site is for blood vessels and bronchi and lymphatic vessels and nerves. Your left lung is separated by a superior and an inferior lobe, separated into a superior and inferior lobe, and the structure that separates those is called the oblique fissure. Remember that the left lung only has two lobes. It's smaller because the heart takes up a big portion of that space. The area where the heart is sitting on the medial aspect of the lung is called the cardiac notch. The right lung is separated into three lobes, superior, middle, and inferior. And the superior and middle lobes are separated by the horizontal fissure, and the middle and inferior ones are separated also by a oblique fissure. So pointing out all of those structures that I was just talking about. And looking at a picture of a lung from looking at a picture of the lung from the medial view of it. And then looking at a cross-sectional view, so you get the idea that the heart is not sitting square in the middle, as we've already learned. It's sitting over more to the left. So each lobe is further divided into segments. Those are called the bronchopulmonary segments. So you have 10 of those on the right and 8 to 10 on the left, and they are separated by little walls called septa made of connective tissue. So each segment is served with its own artery and vein and bronchus. So if one segment is diseased, it can be removed individually. So you may have heard of somebody having a part of their lung removed, and what they do is they're removing one individual segment. Sometimes they have to end up removing all the segments in one lobe as well. The lobules are the smallest subdivisions that are visible to the naked eye. They are hexagonal segments that are served by the bronchioles and their branches. The lungs are most commonly composed of alveoli. The rest of it has some stroma, which is elastic connective tissue. The lungs are very spongy. They are very elastic. If we took them out of your chest and spread them out, they would have the surface area of a tennis court. So there is the whole branching that you see. There's a lot of branching, and so they're showing you the three different segments in the right superior lobe, and the right middle lobe has two segments, and the right inferior lobe has five segments. And so they're showing you all of those different branches, and you can see you have fewer segments over on the left-hand side. The lung is perfused by two circulatory systems. Pulmonary circulation we have already learned about. This is when the pulmonary arteries are taking or bringing the venous blood, the used blood from the heart to the lungs to reoxygenate it, and they branch profusely that feed into those pulmonary capillary networks. The pulmonary veins are what carried it back to the heart. So you had right and left. Remember, you had four of those. Remember that pulmonary circulation was under low pressure. It was high volume, had the same volume, but it was under low pressure, so we didn't damage those very delicate capillaries during the exchange process. The lung capillary endothelium had lots of enzymes that act on different substances in the blood. For example, one that we talked about was angiotensin converting enzyme, or ACE. That is what converted A1 into A2. So ACE is found in lung tissue. Then you had the bronchial circulation. So the bronchial arteries are what are providing oxygenated blood to the lung tissue itself. So the lungs are exchanging gases, but the lungs also have to have a blood supply. Just like we've talked about, a lot of other different organs in the body have to have a blood supply to do their job. So these arise from the aorta and they enter the lungs at the hilum. They are part of systemic circulation, not part of pulmonary circulation. So the pressure is high and the volume is low. They supply all the lung tissue except for the alveoli. The bronchial veins anastomoses with the pulmonary veins, and the pulmonary veins carry most of the venous blood back to the heart. Innervation of the lungs. They are innervated by the sympathetic and the parasympathetic motor fibers, as well as some visceral sensory fibers. The nerves enter through a structure called the pulmonary plexus on the lung root, and they run along the bronchial tubes and the blood vessels. Parasympathetic fibers cause bronchoconstriction, or making the lumen smaller, and sympathetic fibers cause bronchodilation, which makes the lumen larger. So just think about that. In fight or flight, obviously you need to get more air in so you can run from the big giant bear that's chasing you. And so you want the bronchial tubes to be as wide as you possibly can to facilitate that. The pleura, we've talked about the pleura all the way back since the beginning of Bio 210. This is a thin, double-layered serosal membrane that divides the thoracic cavity into two pleural compartments and a mediastinal area. The parietal pleura is what lines the thoracic wall and it on the superior face of the diaphragm, around the heart and between the lungs, and the visceral pleura is what's actually on the lung surface itself. Sitting in between those two is the pleural fluid sitting in the pleural cavity, and that's what allows the two layers to slide by each other. So it provides lubrication and surface tension that allows for expansion and recoil of the lungs. So showing you that cross-sectional transverse section again. Pleurisy is inflammation of the pleura that is often associated with pneumonia. So the inflamed pleura becomes very rough, like sandpaper, and it feels like somebody's stabbing you every single time you breathe with an ice pick in the chest area. So sometimes people are not aware that they have pneumonia. So if they tell you that they're having sharp stabbing pains in their rib cage, you might want to check them. A lot of times older people, their lungs don't work as well as they used to, so they're not aware that they may have pneumonia. The pleura may produce excessive amounts of fluid, which put pressure on the lungs, and makes it harder for them to breathe. Other fluids can accumulate in the pleural cavity. Blood is one of those that happens when you have damaged blood vessels and the blood is leaking out. Blood filtrate, which is the watery fluid that oozes from the lung capillaries when you have left side heart failure. Pleural effusion is any fluid accumulation in the pleural cavity. All right, that's it for part A. I'll see you for part B.